Very good evening to all the young aspirants for CPGET 2025 and also the most important personality and respected resourceful person, Dr. Rajani Madam Garu, Associate Professor, Head Department of Botany, Food and Nutrition, Raja Bahadur Venkatram Reddy, Women's College, Autonomous, Hyderabad. So, Madam is going to explain about the most important topic in microbiology, that is the bacteria. Previously, Madam has clearly explained about the structure, types of the bacteria based on the flagella, based on the types of the nutrition, based on the other important characteristic feature. So in this regard, Madam today is going to continue the topic in bacteria. That is most important topic, that is reproduction. Actually, bacteria shows the two types of the reproduction, that is asexual reproduction, and at the same time the another type of reproduction, that is sexual reproduction. Competitive exam lo manam rayalsindi actually sexual reproduction is absent in bacteria, but the very closer or similar reproduction, that is sexual reproduction, is present in bacteria. That is the recombinations, that is sexual reproduction, that is very related to recombination. So that's why we call that is also treated as sexual reproduction. Three types of the sexual reproduction Madam is going to explain today's class. Madam, please welcome and start your session. So most important bit today will going to appear in our competitive examination. Asexual reproduction through binary fission or the spore formation or endospore formation. The other type of the reproduction, that is sexual reproduction, the transformation, transduction and conjugation type of the reproduction will going to explain today's class our respected Madam. Madam, please welcome and start your session.
Yeah, thank you, sir. Good evening, students. Yesterday class, I'll start to the bacteria. What is the bacteria structure and what is type of photosynthesis of the bacteria? And next one, how to fix the nitrogen? What is the structure and what is the size? These are things are yesterday class we will discuss. Now today we will discuss about to the class reproduction in bacteria. Okay, bacteria reproduce mainly by the asexual method, and therefore they have a dominant haploid phase. What type of reproduction is there? Mainly asexual reproduction. This asexual reproduction, therefore, they have a dominant haploid phase in their life cycle. They do not have sex organs or gametes, but they definitely show genetic recombination is there. Here, no sexual reproduction, no gametes are there, no sex organs, but they definitely show one genetical recombination. Here, the meaning of genetical recombination, that is exchange of the genetic material. What is the genetic material in bacteria? What is the genetic material?
DNA.
DNA is the genetic material. See, here I'll write down the three types of reproduction. No, first one here, asexual reproduction. I'll explain to the asexual reproduction. After that one, vegetative, budding. These are fragmentations and these are things I'll explain. First one is the asexual reproduction. Here, asexual reproduction, main important binary fusion. 1 second, I'll show it the diagram. This one. Asexual reproduction in bacteria takes place by following different types are there. The first method is called the binary fusion. It is the simplest and most common method of multiplication in bacteria. Very most simple and most common. This type is what happened here. The bacteria is the very much multiplications are present. Under favorable condition, the cell divides into two daughter cells. You can observe the picture. The cell divides in two daughter cells by a transfer wall. What type of division here? Transfer wall. During fusion, the cell elongates and there is division of the nuclear material. Which one is the division is there here? Nuclear material in gram-negative bacteria. Yesterday class, I will explain now what is the gram-negative bacteria, what is the gram-positive, what color to reflect. These are things in the plasma membrane to reflect the negative is different color, positive is different color. Here in gram-negative bacteria, it is followed by the development of a simple median constrictions that finally result in the complete separation of two daughter cells without cross wall formation here. Without cross wall formation in gram-negative bacteria, however, first a transfer cell membrane is laid down between two nucleids of a dividing cells, and then the centripetal growth in between that one centripetal growth is present. This centripetal growth of the cell wall from a complete cross wall formation. This transfer wall between two daughter cells, it is very often incomplete. The daughter cells remain connected at the transfer membrane. When the planes of the successive division are parallel, long chain, maybe sometimes this one is the one another sheet and pack the cells are to form. This total process of binary fission is very rapid. Bacteria cells may undergo to the fission every 20 to 30 minutes. Every 20 to 30 minutes and under favorable conditions within 6 hours. Under favorable condition within 6 hours, approximately 250,000 cells may be formed from a single bacterium. This one is that to produce the within 20 to 30 minutes, within 20 to 30 minutes, after 6 hours, you can observe the bacteria number 250,000 cell may be formed from a single bacterium. But it in nature, the essential nutrients are generally a limiting factor. You know that one, yes or no? Moreover, the accumulated waste product of bacteria also restrict their rapid growth. Here, normally growth and multiplication is very fast in the earlier stages, reaching at the optimum and then declining rapidly. This is in this binary fission is different from normal mitotic division. You know what is the normal mitotic, but this is the different from the normal mitotic division as it does not involve spindle formation during the division of nuclear material. Why? In the chromosome is there in this one, but it is not here. Spindle formation is totally absent. And next one is that it is not definitely known as whether in fusion only for the genetic material is equal distribution between the two daughter cells. In this one only you can observe that one genetic material to distribution for the both daughter cells. This binary fusion is characteristic of all type of bacteria, and that is why they have been placed in the seisomycetes. Seisomycetes meaning split or cut. Mycetes meaning, you know, fungi. And split cut of fusion of fungi. That's why this is the under the seisomycetes, you can observe this binary fusion. Coming to the next one, this is a binary fusion. How to reproduction are there? This picture. See, after division, how to bacteria to increase within the 20 to 30 minutes. This is the increase of the bacteria. Coming to the next one.
Madam, excuse me.
Sir,
Actually, the bacteria, Escherichia coli definitely takes time. One bacteria, parent bacteria cell is going to produce two new daughter cells exactly 20 minutes. It is
Yes.
pakka. So, my dear friends, remember, based on the types of the bacteria, it's differ from one species to another species. But Escherichia coli bacteria, definitely we can say 20 minutes only. Okay, madam, please proceed.
1 second, sir. Next one is the budding.